Today's guest is Dr. Tanya Mongan, Clinical Associate Professor and Consultant in Gastroenterology within the University of Nottingham. Put simply, Tanya is a gut health expert, a practicing clinician, and she has particular expertise within the context of the gut-brain axis, and she is specifically interested in deciphering the mechanisms of something we've spoke about a lot on this podcast since we got started, fecal microbiota transplantation, also known as FMT. Tanya has international collaborations that seek to decipher the mechanisms of FMT, and we dug deep into how she feels about the therapy based on the current evidence today, whether she uses it in her clinical practice, and also what the future looks like for patients, particularly with inflammatory bowel disease. Tanya is also pioneering work into something called microRNA-based drugs, and she is specifically interested in utilizing these drugs for the treatment of diseases linked to gut microbiome dysbiosis, including inflammatory bowel disease, cystic fibrosis, C. difficile infection, and maybe even neurodegenerative disease. Tanya is an amazing medical educator and communicator. So you're going to feel a lot of energy from this podcast. You can sense her passion, and it's clear that she is a true expert. Really appreciate all the support that the listeners have been giving the podcast over the past couple of weeks since we got going again. Thank you. This is an amazing episode. I'm sure you'll enjoy it. With that, I urge you to trust your gut, focus on your gut health, and learn as much as you can from Tanya. Why don't we start then with something that I know you have expertise in talking about, which is, has the microbiome bubble burst?
Always a good thing to start with, not.
Right into the deep end.
Right into the deep drowning end. No pressure then. So, has it burst? I mean, it depends who you're talking to.
I did say this was going to be fun.
Yeah. So, I mean, I guess if you're talking to, a microbiome audience, saying it might not go down very well. But I think that, you take it with a pinch of salt. I mean, yes, to some extent it has, because as you and I know, lots have been written in the literature and in the media, about microbiome being basically the answer to everything in life, when in actual fact, we know that's absolutely not true. And I think there's, been a lot of myths and misconceptions, especially in the media, and people basically read things on the internet and they suddenly believe that, if they take a pre or a probiotic, or God forbid, DIY fecal transplants, which, as you and I know, is certainly the case still on YouTube, that's going to fix their acute and chronic health conditions, when actually that's not the case. And I think there's just a greater understanding and appreciation now that the microbiome is incredibly complex and it needs to be very tightly regulated. And so it's good to see some of these consensus guidelines and statements coming through, really calling out these, myths and sort of pseudoscience, which is great, but it still hasn't fully permeated to the greater public, unfortunately. And so, as a clinician, I still see a lot of people coming into clinic asking me which pre or probiotic should they take or which diet they should, follow. And some of them do actually spend quite a good bit of money getting their microbiome sequenced. And that's a concern because, we know that that's just a snapshot on one day. But the next day they could have a salmonella infection, and then it's different. And then the next day something else has happened. They could be on a keto diet. So I kind of have to burst their bubble a bit to some extent as well by saying I don't want to, upset you at the fact that you've spent £500 or £600 on that test, but that really isn't a good measure of your current health status. And I think they get a little bit of upset that they were pulled into that sort of pseudoscience aspect of it as well, and you kind of have to reset their expectations. That can be quite difficult. But for patients, I feel sorry for them.
When they come in with a sort of 16S sequence result, I guess they're coming to you with this hoping that you're going to be able to just tell them everything about their symptoms. But what can you actually determine as an expert when you look at these things, if anything?
Yeah. I think very little, because we don't even know really how the sample was handled initially and what way the person provided the sample to them. And there's just so many variables and so much heterogeneity. And again, we don't know enough about these companies. I tend to just say, actually, I really can't look at that data and tell you anything meaningful. I normally just start with very basic investigations, especially if someone's come in with, very commonly change in bowel habit or tummy pain or bloating. We're going to go and exclude the very obvious things initially and not even go near the microbiome. We'll talk about, often their diet. We'll often talk about, what medications they might be on and all of these other variables that obviously affect their health status. What's their pre-existing medical history? So we start with the medical history, as you know, take good, clear case history, and then figure out after that what are the basic investigations we want to do. Those would not entail microbiome diagnostics at any point, really, outside of a clinical trial research.
And if I could maybe just push a little bit, I know we're just starting, but do you believe that in the future, the microbiome testing component might form part of your diagnostic workup? Like, how optimistic are you about the technologies we have and the future in terms of microbiome sequencing?
I mean, I would be reasonably hopeful that we will get there, but it will have to come through a very regulated, rigorous process, much like how FMT and live biotherapeutic products are being regulated now currently. So we need to have these standardized, harmonized approaches so that, maybe we're looking at a centralized sequencing supplier that's affiliated to the NHS, for instance. If we've got something that's harmonized across all the hospitals and it's standardized, then we know that we can probably rely on that information, and also the protocols that go into acquiring the samples and analyzing the samples and then the data interpretation around that. So I think we would need as NHS operators control over those aspects to make sure that it's all very harmonized. If that was the case, then I think it could be theoretically helpful, although from a snapshot perspective, maybe not. Maybe we need the longitudinal data.
So it sounds like there's maybe two aspects that you could potentially elaborate on a little bit more. One is that, as you say, it's just a snapshot. It's just a single day, and it's this evolving ecosystem, and that's one piece maybe to unpack. And the other, you touched on it a couple of times, was different providers, different labs.
Yes.
Maybe you could just elaborate a little bit more for the listener about why that potentially is a problem.
I think the problem is that it's a bit like running FMT studies across multiple sites. If you don't have protocols that are fully harmonized in terms of how you go about collecting the sample, how you go about storing the sample, then how you go about actually processing the sample, and then you get finally to the sequencing step, and then beyond the sequencing step, the tools that you use to analyze the sequencing data. So there's just a real hurdle of things to gallop over. You'd need to be a pretty good stallion to get through all of those hoops without making a mistake. So I think that's partly one of the problems with microbiome science, has been all of those difficult areas. And then you might think, actually all these kits are the same, these diagnostic kits are the same, but as you and I know, they're not. And some of the reagents are contaminated as well. So there's this whole microbiome contaminomics field.
I've not heard that one before, but I like it.
It's so cool. And people actually identifying common microbial species in some of the reagents of the sequencing kits. And so that is actually something to be aware of as well. So there are baseline microbial species that maybe they're already in certain reagents. So you have to strip them out rather than associating them with a particular disease state from the outset. So there's those diagnostic conundrums, even upstream how you take the DNA out of the sample. Again, there's so much heterogeneity across the DNA kits. From one kit to the next. Some of the meta-analyses were done over the last 5 or 6 years have shown that. You can get 100-fold difference in DNA out between different kits. I mean, that's insane and concerning.
So it is concerning. So if everyone's using different protocols, different reagents, maybe even different sequencing technologies, because there's a few, isn't there? Then each of those variables could be having an impact on the end product.
Yes.
So you can't compare them all elegantly, I think is the key takeaway there. So if a patient comes to you and says, Prof, should I be getting my microbiome sequenced?
Yes.
What is your advice and recommendation on that one, if anything, at this stage?
Normally, I would say to them, because they're super keen. Some of these patients are super keen, very well read. They just want to, get into the midst of it. And those patients, I have to take an extra little bit of time because I have to kind of take them back to the beginning. Normally, I would say I wouldn't do it. Unless they want to be part of a research study or a clinical trial, in which case things are a little bit more harmonized, clearly. But outside of a clinical trial or a research study where we've got really good controls in place, I would typically say for them not to do that because we're just not there yet. We haven't got to that point where we can integrate microbiome data into everyday clinical practice.
Yeah. And then just one final point on this then. The snapshot.
Yes.
Could you just elaborate a little bit more about what that means and, how things are dynamic? And you mentioned salmonella,
Yeah. So what I've tried to say to patients, I try to be quite simple with them because they want some take-home messages, simple take-home messages. So normally what I would say is, in any given week, the microbiome can be highly dynamic and change over the course of a few hours, even days. Even the time of day that you provide a sample could theoretically have an impact on the composition of the microbiome. So that's been shown to be the case and published not that long ago in Nature. So there's so many variables across the week. Their diet will change across the week. Maybe the medications they're taking might change across the week. They may be inadvertently taking extra fermented foods in the diet, and that will obviously change the makeup of the molecules in the stool. So there's lots of different things, even how much alcohol they might consume across the week. So there's lots of different things that people don't fully realize could theoretically impact on their day-to-day microbiome composition. Plus also there may be terrible stressors, environmental things coming through. And again, that also could theoretically have an impact. So when you say all this to them in clinic, they're like, what? They're like, well, what's the point of this microbiome then? So I think they realize then that the true complexity of it. And then they come back to basics. So what blood test do you want?
Yeah. When you describe it like that, I had lots of things going through my mind there. I was thinking, fecal microbiota transplantation donors, that means that they have that much variability as well, which is a challenge that we all face.
Yeah.
So I think the key takeaway is it might be interesting if you can afford it for your own information.
Exactly.
But it doesn't help you as an expert and a clinician in the diagnosis of the treatment at this stage today. Yeah.
Correct.
That's fair.
I think so. Yeah.
And just going back then to the big question, has the microbiome bubble burst? Could you maybe take us back to when you first got interested in this space? And where were we on that hype curve? Were we kind of just creeping up? Was it peak interest, or did no one know anything about the microbiome at that time?
Yeah, interesting question. I think I probably got interested in it maybe about 10 years back. So certainly around sort of the time I was handing in my PhD, I was, looking very closely at microbiome in the context of C. diff, because it's obviously the exemplar state of creating a dysbiosis. But my PhD was very much immunologically based. It was very molecular. It was very sort of humoral immunity to the C. diff toxins, for instance. But I realized at that point that, separating the immunology out from all these other omics was not a good idea, even though you would spend 3 or 4 or more years doing a very deep dive into the immunology of something. In actual fact, if you integrated all these other data sets, you could probably create more interesting findings, at least as far as pathogenesis is concerned, and obviously potentially identifying new targets for therapies. So I kind of was a bit upset with myself on handing in the PhD. It's like, really? I should have done some microbiome work before now. But actually, the curve has only been coming up in the last particularly last 6 to 8 years, although all the sequencing came into power, of course, in 2004 with, Venter's work on sequencing marine bacteria, which was just beautiful work. And, and then basically applying metagenomics to uncultured organisms. That was all 2004, hard to believe. So not that long ago. Did they start to apply metagenomics to these bugs? And then, since then, obviously, then you've got the first RCT for fecal transplants in 2013. And I think really since 2013, that's when it's gone crazy. And that would have been the time I was handing in, thinking, there you go. And then this all happened. So actually I shouldn't feel too guilty that, I wasn't on the pulse, so to speak. But since then, it's just been incredible. It's insane. It's a bit like the popularity of the internet. You're like, what did you do before the internet? Yeah. It's just not right.
10 years ago was when, roughly 10 years ago was when I read the New England Journal of Medicine paper, Van Nude.
Yes.
And that changed things, I guess, for me as well, because I was like, sorry, what? I couldn't believe that they were actually doing this. And then the data looked amazing.
Yeah.
So for an expert in C. diff, which you are, what was your reaction then? Because you'd done your PhD on C. diff.
Yeah.
What was known before, and how did this change things?
I was just completely delighted by it, because I just thought, hang on a minute, this is totally different paradigm. This is a paradigm shift here. You know, they're using C. diff obviously as an exemplar state, but they can actually reverse this dysbiotic disease with this FMT. I mean, at the start, it was kind of like a yuck factor, but the yuck factor to me lasted about 2 seconds, 2 microseconds. I was like, hang on, how does this thing work? And at that time, we weren't doing an awful lot of FMT. Obviously, it was coming into its own, wasn't it?
Absolutely,
so,
yeah.
You know, we had to wait another sort of 3 to 4 years for the other FMT trials to start coming through, you know, the fresh versus frozen, and then the colonoscopy versus capsule. Exactly. Yep. So we had to wait for all that. So in around 2017 or 2018, that's when I started to collaborate with people that were doing a lot of FMT, especially in Canada, where some of the big FMT trials were really coming on par. And there was a chap there called Tom Louis in Calgary, and a colleague of mine at Alberta, Adina Gao. These guys were coming along and doing, you know, really interesting sort of innovative work with FMT, particularly around comparing the different ways of transplanting subjects and looking at the comparisons. Right. So actually, I'd actually made a point of meeting them at one of the Keystone Symposias in Banff, of all places. I thought, hang on, you know, I'm going to make a holiday out of this. I'm going to firstly meet these collaborators.
Sorry, can I just say for listener, there are 2 Banffs.
Yes.
One is the stunning, picturesque Canada, but there's another one in Scotland, just FYI.
Oh, right. Hello.
which I will send you.
Yes, please tell me.
I will send you the details of Banff in Scotland. It's a little bit less exciting, I have to say.
Good to know. Noted.
Just FYI.
FYI, people, this is the Banff in Canada.
I'll just tell you, the reason why I know this is because a few months ago, I was having dinner with a Canadian, similar age, going through similar kind of life events. And I said, so, you know, where would you like to go with your wife? He said, Banff. It's amazing. I was like,
which one?
No, I didn't know about the other one in Canada. So I was like, sorry, what? I was like, Banff. I was like, you're coming all the way over from Canada to Scotland to go to Banff. I was like, that's so interesting but weird. He was like, no, this is Banff. And then he showed me, it just looked unbelievably epic.
It is epic. It's like some kind of crazy epic. Yeah, it is so good. And to be honest with you, at the time, it was minus 25 when I went over. And I just remember my colleague saying to me, my collaborators, you know, Tanya, we've got a special coat and boots for you, so you don't really need to worry about bringing your Irish version. Just bring, you know, we'll sort you out. I remember landing in Calgary thinking, you know, first thing I saw was all these people wearing cowboy hats, and I thought, oh, this is a great place. Because I had to get to the airport, obviously flew into Calgary, and then my colleagues lifted me up, and honestly, minus 25, and I thought, you know what, this FMT is worth it. Yes, it's minus 25 with about 2 meters worth of snow. But that was a really good meeting, because that meeting really started to introduce FMT. It was a very microbiome driven symposia. And, you know, after that, then I really started to get into it, because, you know, with these good collaborations, you can access clinical trial samples. Right. And at that time, there wasn't an awful lot of people doing much mechanistic work inside the microbiome space. It was very much, you know, related to compositional changes, but only starting to look at that. Not everything else has gone since then.
So I guess this is a really big question, but do we know how FMT works?
I think we're only really scratching the surface, honestly. I really do. I mean, I think at the minute, you know, everybody tries to give a little bit, you know, try to contribute to this area, but at the minute, you know, the host is so complex, the microbiome is so complex, then you start to look at host microbe interactions, and then you just, you know, you have an aneurysm.
Yeah.
So it's very complicated.
Someone once said to me that it could be the most complicated thing in the universe, particularly if you consider the brain, because it's just, there's so many moving parts.
I know. Well, there are lots of analogies, aren't there? There's like this dark matter analogy around it, and people like to use all these cosmology comparisons. I mean, I think it is incredibly complex, and this is part of the problem. If we knew how it worked, we could have all sorts of treatments and precision therapeutics. So I do think that we should, really, really dissect the mechanisms. And I think a lot of people are doing that. I mean, a lot of people looked at the microbial changes and diversity and richness, and we see that improves with transplant. We've seen a lot of work around metabolic changes and short chain fatty acids and bile acids and how they're important. But really, some of the other aspects are absolutely not looked at, hardly. Giant research gaps. I mean, giant. Around the immune side, what's happening immunologically in the blood and in the tissues, and what's happening at more proximal states, like epigenetic changes. So we've been trying to look at those latter aspects because we think those are the most neglected at the moment. And also sometimes you want to do something different. You don't always want to go down the microbial metabolic route. You want to sort of try to look at something a little bit more upstream potentially of those things, because all of those things have to do something to the host immune system. You get the microbes changing, then they have to secrete the metabolites. Metabolites just don't go off on holiday. The metabolites actually do affect the immune system. So it's those immune changes that's really interesting.
So could you maybe take us back to when you first started to work with Dr. Louis and Dr. Cow? What was known then, if anything, and then like how our knowledge has accumulated and built, and the work you've been involved with, between then and now?
I think back in 2013, those were pivotal foundational trials, but they collected samples for sure, and they were very sensible about doing that. In fact, they were some of the first investigators to collect samples for mechanistic work. But they weren't really funds and how would you say, funders that were really interested in funding that type of research. And I think what's happened in recent times is you've got a greater emphasis around the mechanistic changes associated with the clinical trials. Before, it was just the clinical trial. What happened? Is it effective or is it not effective? But then later people started to really collect the samples longitudinally following transplant, and then to start looking at all these microbial, metabolic, immunological changes that are occurring. But in 2013, that wasn't the case. People just wanted to know, does this thing work for C. diff? Then in the decade that followed, people started thinking, actually, we need to start really dissecting these samples now. Where are the changes? And the focus was initially on the microbial changes. I mean, that's the lowest hanging fruit. That makes sense. Look at the microbial changes. And there were some commonalities and consistencies, but because there were a lot of different trials going on, not all of them had harmonized protocols, and there's so much heterogeneity meant that there was no absolute consistencies across the board. But people started to look then at the metabolites, targeted ones, especially short chain fatty acids and bile acids. But only in recent times, as I say, as people really start looking at the immunological and sort of epigenetic side. I think that's largely because the tools of the trade are improving as well. Sequencing is one thing, but then metabolomics has come a long way, and all these different spatial transcriptomics and what have you has come a tremendous way as well. So, I think the multiomics has helped develop the field, those technologies.
So as the cost has come down and the throughput's increased, Yeah,
I think so. And things are improving. Suppliers are better with these multiomics now. I think the gap still exists with regards to data integration, because still there aren't that many really good computational biologists around that can handle these complex data sets. So you've got that integration issue still a problem, and training problem. There's not many people that can do it. And then you've got, I guess, some of the problems with data interpretation, because you don't have everything, we don't have all these reference databases that we still aren't fully properly annotated. And it's good. It's not bad for microbiome, not bad for bacterium, but it's not so good for virome, for instance. Not so good for all the fungi. So this is why we still struggle a little bit with the analysis. We don't want to overinterpret, but at the end of the day, it's not that difficult to overinterpret sometimes.
Maybe you could just expand on that a little bit then. So how much more do you think we know about the changes with the bacterial components in FMT versus the virus groups and the fungi, and how important do you think they are, the virome and the microbiome?
Yeah, I mean, I think the main emphasis, of course, has been on the bacterium, isn't that right? So the bacterial shifts and diversity and richness shifts, and they seem to all sort of theoretically improve on paper, certainly, and then in clinical sense, they do also improve, but not absolutely 100% of the time, but in quite a few clinical studies and, of course, in animal studies as well. I think the difficulty is that, you'll never seek perfection in this reign. There will be variability across the board. That's just part and parcel of the problem. But I do think that things have certainly improved as far as, reproducibility of data because of the improved protocols and the greater recognition that we just need to be all on the same page with how we handle samples and process samples, etc. So, I think things are getting better, but we're still nowhere near where we should be. But we will be probably in not 10 years' time.
Right, because the technologies will continue to get better.
The technologies will continue to improve. The issue for us is that bacteria is only one strand. We're dealing with the trans kingdom, aren't we? So we've got microbes, they're important, but the microbes just don't consist, obviously, of the bacteria. We have to look at the parasites. We have to look at the viruses. The difficulty there is, sequencing the virome is quite tricky because there's different ways of doing it. And even then, it's really not well annotated compared to the bacterium. So the bacteria side, better annotations, you know what you're identifying, but not always. You still can't culture everything, but you have a fair good idea. You can map certain things out there. The mapping in the virome is much less mature, and there's room for, really making mistakes. So that's where you shouldn't really overly interpret. The other side of the coin, the fungi is the least well researched as far as the microbiome is concerned. So that's really difficult, again, because of the way we don't really know much about it.
Right.
So then to annotate and map it, that's what we need to try and do.
Is map all the kingdom in all of its different respects.
Map the whole trans kingdom, yeah.
And if you were to give a kind of impression based on your expertise as to the relative contributions of each, would you say that you think the bacterial components are most important because they're metabolically active, or the viruses are maybe more important because they infect the bacteria in the case of phage? What's your expert impression at this stage based on everything we know and understand at the moment?
It's difficult. I would probably say bacteria are very important. So my colleagues and I, we finished a clinical trial in Canada recently. We presented at UEG looking at lyophilized FMT. So that's FMT with everything in it, bugs included, versus a lyophilized sterile fecal filtrate. So that's no bugs, just the filtrate. So with all the little small molecules. And, in the context of the whole lyophilized FMT, that was 87% effective at 8 weeks at preventing recurrent C. diff versus 65% for lyophilized fecal filtrate. So that tells us that microbes are important, but it also tells us that fecal filtrate's doing something in 2/3 of the patients.
Yeah, that's really interesting.
So the bugs are important, and you probably do need the bugs to get the best clinical efficacy. That being said, 2/3 of patients were responding to the filtrate, and the filtrate has no bacteria, no live bacteria. It just has the filtrate, the small molecules. So inside that small molecule soup, though, is clearly lots of ingredients that are helping the host, shift this bug. Or prevent this bug, and you can speculate till you die, but basically we don't really know what these small molecules are. We can speculate, that they're metabolites, obviously, and we can speculate that they may be antimicrobial peptides. And we can speculate
phage, maybe the phage as well.
Definitely phage, because even when you filter to create the filtrate, you have to put it through a little tiny fine filter, 0.2 micron. When you put it through that, some of the phage are actually smaller than that 0.2 micron. So you might not necessarily, some of the phage are tiny. So,
Wow.
if they're really small phage, they're going to get into that filtrate. And so, yes, we expect that phage will be in there alongside the small molecules like the metabolites, alongside the antimicrobial peptides and probably the microRNAs, because the microRNAs are really small. So
Wow.
I think that will be some of the constituents of the filtrate, but there may be other things that we're not familiar with.
Because I think this sounds like really interesting work that I'd love to read. It builds on, I remember a very small study from a group in Germany, I think maybe 5 years ago, that showed that N=3 sterile fecal filtrate seemed to have an effect on C. diff.
Yeah.
And then that triggered this big debate around, will you even need the bugs at all? It sounds like that's been answered, at least in part, by this work.
Yes.
But there could be more in the soup.
There could be more in the soup. So this is why we have to keep thinking, yes, we've answered the question that if you compare them head to head, the bugs seem to do better than the soup. However, the soup is still working for some people. So that means that we need to go back to basics to try to really understand and dissect out what's in the filtrate. I mean, even if you take it a step back and say, what's in the poo? Well, I mean, that is real dark matter. Because, we know that there's colonocytes and mucus and secretory immunoglobulins. We know there's lots of microbes there. We know there's lots of metabolites there. But there's also other things recent times that I discovered. So if of interest, there was a paper in 2015 in Science and Technology of the Environment, I think it was, by a group, I think, in the US, who basically were doing a lot of wastewater studies and essentially said that in a city of a city of 1 million inhabitants, they were flushing down the toilet $13 million worth of precious metals, including gold. So I presented this at the BSG last week. So at the BSG last week, I was saying, okay, so this is what's in your poo: colonocytes, mucus, small molecules. But just in case you really, want to think about it as another liquid asset, here is what else is in the poo: precious metals, including gold. Yeah. So it's insane. So, it's a resource that's been neglected in that sense. Of course, you would need a very big city to get out millions and millions of pounds worth of gold, but it goes to show you this could be my next job. It's like so there's a lot of different things in poo, and this is the thing, you see, it's trying to understand what are all those constituents, and then that's just what's in the poo, but then you strip out the bacteria, you see, and then what are all these other little tiny things. We probably don't have the technology to fully annotate, you see, yet.
No, probably not. And I also read a long time ago that if you're a smoker, then there's some ash in there as well. It somehow finds its way down there. Finds its way. Very, very interesting. So basically, the key takeaway so far is there's still a lot we don't know.
Yes.
What was the reaction, by the way, at BSG?
Well, I think there's a few people smirked, probably thinking I'm pulling their leg, but I said, no, here's the paper. I was saying, I do wastewater stuff. Had I known about this, I would have been on this. So, I think it came as a little bit of surprise to some people. Well, especially when you show them a gold toilet, actual gold toilet. They're like, oh, come on.
Wow.
But
an actual gold toilet. Oh, that was part of the joke. Sorry,
that was part of the joke. Here is a gold toilet. It was basically, you know what it was, James? It was basically, forget the Bitcoin. This is the new cryptocurrency. That was literally it.
Oh, it's so funny. I'd love to see that, actually. That's really, really, really funny. I've been talking about stool and poo and FMT for the last 10 years. And I've heard of liquid gold, but no one's actually ever said there might be trace elements in there, which is super, super interesting.
I know.
Wonder if there's a venture in there for somebody very entrepreneurial.
I am thinking, listen, I do wastewater studies all over the place. I'm thinking this is a new arm. I'm just going to have to work out the processing, technology.
You just need to figure out how do you extract the minute particles. There'll be a way. Oh
no, there's ways. There's ways, because people do.
Don't disclose if it's a secret. You need to patent it.
It's not patented. There are chemical precipitation methods of doing it. It's
Oh my
just that,
goodness.
if I took this wastewater into the lab and started trying to do this, I think the biosafety committee may have a few things to say.
I'm sure they would, but it's interesting if it's just being flushed away anyways.
Yeah.
I mean, maybe, okay, interesting. All right, making a note, next company. Anyways, no, I'm too busy. Back to the wonderful world of FMT then. So what is your research kind of focus on at the moment then? If you could explain to the listener, what are you excited about and what are you up to?
So part of my research is around FMT mechanisms. So really taking it back to, the black box bit again and trying to sort of reverse engineer how fecal transplants work in a nutshell. So one of the ways we've been doing this is trying to look at some of the small molecules that are in the poo pre and post transplant, trying to see what's upregulated and what could theoretically therefore be, a therapeutic solution, a precision therapeutic solution. And so we rather than look at microbes and metabolites, we thought, we will look at these other small molecules instead. So we'll look at these microRNAs. So these are basically the, genome master regulators, essentially. So they regulate gene expression. They're really small molecules. And
so what are these microRNAs then for the listener? Sorry, you mentioned they're tiny and
they're tiny. They're only about 22, 28 nucleotides in length. They're relatively small. They basically are master regulators of gene expression. Very small RNA molecules that are master regulators of gene expression. So if you've got a certain set of microRNAs, if they're upregulated, they will repress gene expression. So that's what they do. When you upregulate the microRNA, they repress the gene. So really what you want to do is try to identify these pairings. So what genes are pairing with the microRNAs. And essentially, after quite a lot of work, we were able to detect certain microRNAs that are upregulated following successful transplants into human clinical trials of FMT in Canada, because we needed the discovery and sort of the validation cohort. And we were able to show consistent changes in the same microRNAs following transplant. And when we looked at that, then we went back to see what genes they regulated. And interestingly, the genes that they were regulating were inflammatory gene targets, which was really interesting to us.
So the RNA was going up, the inflammatory targets were going
coming down. Exactly. So so in the context of C. diff, there was a deficit of these anti-inflammatory microRNAs. And then in successful transplant, these microRNAs were being restored in the blood and in the mucosa. So in the colonic biopsies and in vivo models, in mouse models. So that to us was like, oh, well, that's quite a consistent finding. Can we now take those microRNAs and see, can we create a therapy around those microRNAs? So,
sorry, before you just go any further, a really silly question, I think, from me. Where do the microRNAs come from?
So these are endogenous. The body makes these microRNAs, but the microRNAs can also be produced or can also be derived from the diet. So you can get dietary microRNAs, exogenous, coming through the diet, and then your body also makes these microRNAs. So there's these fancy enzymes involved in bioengineering the microRNAs in the body. And what's really interesting is they reside in different places. So the microRNAs that we're interested in are epithelial microRNAs.
Got you.
And
interesting.
So, it's really interesting. So basically we decided then, okay, initially, we started to look at the effects of these microRNAs in different cell lines. So naturally, we wanted to look in the gut to see, well, if we take human gut cell lines, can we look at the effects of these microRNAs in vitro? And we were able to show that they were protective against a range of microbial toxins. So all the nasty stuff. So protective against C. diff toxins, protective against heat label E. coli toxin, protective against lots of fungal toxins. And things like LPS, which is inflammatory. So that was really interesting. I thought, okay, good. So we know that they're doing something functionally in the epithelia. Can we just give them to the host and see what will happen? Well, you can't actually give them to a human host, obviously, without all the tests. But even in an animal context, they'll get digested in the stomach with all the juices and the acids. So you have to basically put them into a carrier system. So what you can do is you can create different nano-formulations, tiny micron-sized formulations that will carry these tiny molecules into the cells. And so some of the molecules that we're using are polymers, and lipids. These are tiny lipid and polymer carriers. And you can put the microRNAs inside these and then use the nanocarrier system to bring them into the cells.
Okay, so I think I'm understanding. You're identifying post-successful FMT that these certain microRNAs are associated with potential benefits. They're restored after FMT, and you mechanistically tied it back to inflammation.
Yes.
Then your next challenge was, how do we actually deliver these?
Yes.
and you're doing that through these lipid systems. That's very interesting.
Yeah. So you have to put them inside a lipid system, or you could put them inside a polymeric system. At the minute, we're putting them inside polymeric systems that we're engineering in pharmacy, and those are non-toxic. And you can get them into the cells. So we've seen we can get them into the cells, the gut cells, and we can get them into animal gut cells. And they're non-toxic. And the other thing is we've also looked at them in different organ-on-chip models. So these are like the fancy organoids, but inside a microfluidic system, like a little chip. And you've got a lot more control of the fluid system inside those little chips. So they really are excellent because they're not animal models, number one. So, it's good for, C3RS animal replacement technology, but also it's much more human relevant, physiologically relevant platform. And now the Federal Drug Administration have really embraced these for toxicology studies. So, they're still using animals for toxicology, but increasingly now they're insisting upon the organ-on-chip systems to also come in. And so we were using them. So we've been using colonic organ-on-chips, and we've been using, so basically collonoids inside a chip, and using liver chips from a company called Emulid. So you can buy the chips and then the cells separately, and then you put the cells inside the chips. Or you can create your own cell lines, which we've started to do as well, and mutate those lines and then put them inside the chip, and then you can interact those cells with other cells or other biosamples to make them personalized. So one of the things that we're doing at the minute, one of my students has been developing these personalized gut avatars. So you can basically have the gut cells inside the chip, and then you create a personalized gut avatar by actually coming in with the samples from the patients.
The microbiome samples, the stool samples?
Stool samples, or the blood samples, or immune cell populations from the participant. So you can create these avatar systems, and then this is really personalized now because then that person, let's say it's called Johnny's poo is inside that chip. So that's Johnny's phenotype. So it's really good because then it moves it just from a standard chip to a personalized avatar system. And then you can then start to look at different treatments inside that platform. So it's good for pathogenesis. It's good for identifying therapeutic targets. And it's really good for assessing the efficacy of different treatments. Really good, especially for IBD, because only 2/3 of patients will respond to any one biologic. So you end up using these systems to identify the best treatment for your particular patient. Yeah.
Wow. So are we actually able to do that in clinical practice now? You can look at...
You could theoretically do it in clinical practice now. So one thing we're doing is we've got a cystic fibrosis gut model at the minute. So one of my students has created the cystic fibrosis gut cell line by simply mutating out the gene of interest, the FTR. And then she's put that inside the gut, the microfluidic chip system. And now she's adding plasma and sera from those patients into the chip system to look at, what's happening with their gut barrier and their inflammatory signals. And it's a really nice system. And with that system, any of the really interesting findings, she's then taking it to an in vivo model. And so the in vivo model that we have is the Zebrafish model, because the Zebrafish, again, it's really high throughput system. And it's fantastically good system. It's a totally transparent system. You can see right through the Zebrafish. It's really good at looking at gut changes. So you can look at gut barrier changes inside the Zebrafish. So we've been trying to create avatars with the chips and also the Zebrafish. The patients love it because they're like, hang on a minute. what did you say you're doing with my sample? I'm like, well, I'm putting
Making
your
an
sample
avatar.
into the yolk sac of the zebrafish. And the Cystic Fibrosis Society were really interested in it, so they invited my students to do like a video diary of her
Wow.
experimental work, because I think it's just a little bit off the beaten track of
It is.
you know.
I hadn't heard of this workflow before. I think I heard of gut on a chip many times and appealed, but I've certainly, I think, based on what I understand, it's quite appealing that you're not competing with, for example, in a mouse, the fact that a mouse has a mouse microbiome versus a human microbiome, which is, well, in effect, adapted for a human. So there isn't that issue with the gut on the chip, is there? This is the person's microbiome. So that overcomes that.
Yeah.
Really interesting.
Yeah.
And on the zebrafish, when you say it's sort of translucent, as in you can just look into its GI tract. Yeah.
You can look. So it makes it a wee bit easier to dissect, at the end. But the zebrafish are remarkably resilient. So you can, some of the experiments involve cutting the tail. The tail just grows back. I mean, it's like, it's a fantastic regenerative model. Yeah.
Wow. Okay, cool. And is there still limitations on how you seed the zebrafish microbiome, for example? How do you do that? It's in the water?
So at the minute, we haven't manipulated the microbiome yet, but it does have its own microbiome. At the moment, we're particularly interested in looking at the gut and brain barriers inside the zebrafish. So the gut-brain axis work inside the zebrafish. Want to know if we put the biosamples inside the water they're swimming around in, then they will just take it in.
Okay,
so you don't even have to microinject at this stage, but you could microinject it, but they will take it in the water. Yeah, and then you've got labeled cell lines. So you've got different transgenic cell lines that you can follow the neutrophils and the macrophages inside the zebrafish, and you can see where they swim to after they've consumed these different biosamples.
Fascinating.
So you could do it there. You can do it in flies as well. So we're trying to do it in flies. Flies are very smart, though. They do
Drosophila.
not. Yeah, Drosophila. Flies don't really like eating poo, sadly. Strangely, they won't. They don't really like human poo, but you can. They will take Sarah. That's a little token out there for you. So
Thank you.
So flies are tricky, but zebrafish don't mind.
Interesting. So I think we just about got to the point where you were dosing the microRNAs. Have you got any?
Oh, yes. Yeah.
Yeah. So where are we with that? Because it's interesting. Because it's more, I can see advantages to the microRNA for sure.
Yes.
I don't know how easy they are to manufacture. Maybe that's another consideration.
So at the minute it's quite a multidisciplinary thing, because we work with UCL. So UCL and the School of Pharmacy here in Nottingham, because they've got good experience of polymeric nano-formulations and lipid-based formulations. Of course, some of those are patented as well. So we are going in with their formulations, and then we're testing them in vitro initially. So lots of CACO2 cell transwells, the gut on chip. And we also work with Roisin Owens in Cambridge. So Roisin has a gut immune model. So it's in test epithelial cells with some other monocyte cell lines, for instance. But you could change it up for organoids as well. So we use that system. So those systems we use initially as the in vitro assessment high throughput stage. And then we move to lower throughput stages, which is usually the gut on chip, because it's horrendously expensive. And then after the gut on chip, then we take what falls out as the most effective formulations. Those are then taken to the animal models. So we have got some MRC funding that we got just the end of last year to take it through that sequence to the murine models. So the murine models will be IBD, 3 IBD models, and a C. diff IBD model. So we have a collaborator in Clemson who's an expert on C. diff murine work. She's coming over to develop the C. diff IBD models with us.
Fascinating.
And then that'll be interesting, because they hang out a lot together, as you know. Patients with IBD get a lot of C. diff. So there's no real treatments at the minute for both diseases. The microRNAs are beneficial, though, because our formulations protect against inflammation. They protect the gut barrier from microbial insults, and they're also anti-inflammatory. So even if you get through the barrier, they then dampen the inflammation in the lamina propria.
That's interesting.
Yeah.
So what's your vision then for how far these can go? Indications? Are they standalone treatment, or is it alongside some of the standard care? Have you given some thought to that?
It's difficult. I mean, it's quite speculative at the minute because the RNA therapeutics have come tremendously along in recent years with COVID vaccines. They're all mRNA vaccines, so they've done superbly well. And there's now a lot more interest in RNA therapeutics. So I think that ceiling can be, we can get there with this type of treatment. The microRNA is a slightly different tactical approach, but nonetheless, these are definitely drugs that are coming of age. Especially more interesting when they're FMT regulated as well. So people are starting to think that is really precision medicine. I do think that it's going to take a lot of preclinical work, though, to persuade everybody that there's no really major negative effects, particularly off-target effects, because microRNAs might regulate more than one or two genes. So we need to understand, firstly, what they're targeting in vivo. And once we know what they're targeting in vivo, then we'll be more confident. But so far, the nano-formulations that we've created are completely non-toxic in the liver and gut chips. And they're non-toxic in the animal models, free of infection. So we know in that sense, that's a really good starting point. The next thing now is to see how beneficial they are in the context of an IBD model system.
Yep.
And
if that data is good, then maybe there's a spin-out company.
Yeah, I mean, we would be. Yeah, I think that was probably something that we would do. In Nottingham at the minute, we work. There's another chap I work with, Wang Chang. He's a chemical biologist. So he and I are now. We did have a DPFS to create a synthetic antimicrobial C. diff antibiotic. That's very sort of, how would you say, doesn't impact the gut microbiota like vancorfidoxy, for instance. So it's quite selective. We're moving now to a second DPFS with that drug to try to, really uplift it preclinically to get it to an IND enabling status. I think if we can get that one really up and operational, then the microRNA would come in on the spin out side with it as a secondary agent. Because I don't think any single agent is like you and I know, no one single thing fits everybody.
Nothing's perfect. Nothing's perfect.
No. And it's going to be like it's like IBD all over again, isn't it? Some patients will respond to one type of drug, another set of patients will respond to a different type. So we need at least two or three things.
Absolutely. I mean, people ask me, what's your vision for our drugs and FMT? I say, well, look, I don't think everybody needs an FMT. I think that's just not correct. There's a group of patients who've got, really profound dysbiosis who are probably going to benefit from an FMT more than someone who's maybe earlier on in that journey. And there'll be a role for the precision therapies. There'll be a role for the single strain, the consortia, and there's probably be a role for the full spectrum as well. Yeah. And that's good news for patients.
Definitely.
There's options, and also for physicians as well who are helping. I can really see how you mentioned precision. It's definitely the correct word. You go from this full spectrum ecosystem right the way down to this short chain of nucleotides, and you precisely show how that's having an impact on cells and physiology. I think that's a really nice story. And also, I think a drug, a microbiome sparing antibiotic makes sense. Fidaxomicin, I'm not sure if you use it in your clinical practice, but it's popular now, isn't it?
It's very popular, especially for recurrent disease. We would definitely use it. I mean, we still use, the guidelines are pretty clear. We use Vanc first, but for recurrent disease, we are increasingly using Fidaxi. Yeah.
Interesting. And how does FMT fit into your clinical practice at the moment?
At the minute, it's been now, I can't remember how long we've had it going for now, at least 4 years or so. Even in that time frame, we haven't done, more than about 3 dozen or so, even less. I think the issue is we tend to reserve it obviously for the recurrent cases and really the deeply refractory cases. Not everybody wants to agree to it because they still see that cosmetic issue with it. But for those that do agree with it, it's usually very beneficial. I think the cases that we find difficult are the severe fulminant cases. Then, those are very tricky because they kind of pass the point of no return sometimes. Yeah. But if you can catch patients earlier on, then, it's actually very good. We would typically start with the usual stuff. So Vanc, fidaxomicin. Sometimes with the multi-recurrent cases, we will give immunoglobulin, so polyclonal immunoglobulin. So we are reasonably happy with that treatment. We've audited that now recent times. It's effective in about 50 to 60% of patients.
And in the UK, is it possible to get the Merck monoclonal antibody.
So Bezo is now, it's now no longer. So as of the beginning of this year, Merck have closed down on bezotoximab. So they've closed their program. Right? Yeah, they've closed their program. No real explanation as to why, but they've closed it down. It's now it's a non-entity. It may be partially because maybe it was only picking up in the US. It certainly wasn't a global pickup because it was very expensive. NICE did not approve it in the end. NICE basically didn't. NICE thought it wasn't economically sensible to bring it on.
Wasn't cost effective based on the efficacy.
And my colleagues in Canada weren't using it either, again, for the same reasons.
Interesting.
So, that is very interesting. The clinical trial work originally, though, wasn't unbelievably impressive. But
it didn't excite you.
It didn't excite per se, but it's disappointing to see it go, though. But we weren't ourselves using Bezlo at any point because it wasn't approved for us to use. So that's the reason why we use polyclonal immunoglobulin, because it naturally contains antitoxin antibodies. And we showed that. So we had various assays that we had developed, these microarray assays that you could assess simultaneously IgG, MG, all the subtypes against the toxins. And we could see that the preparations, the different commercial preps, contained different levels of antitoxin antibodies. And so we would select out different preps based on the titers of neutralizing antitoxin antibodies. So
that's really interesting.
You can do that. I mean, to be honest, the polyclonals are not a bad idea. They make sense.
Yeah.
The difficulty
is... Are they a bit like a soup as well, the polyclonals?
It's a plasma. So you need a plasma about 1,000 people, create the polyclonal immunoglobulin. It's all commercial preps, but the difficulty is they're quite restricted. They're not widespread because we need them for common variable immunodeficiency and other immunological deficiency kits. So we have to have 2 consultants sign off on the polyclonal IVIG for C. diff.
Wow.
Yeah, for any condition.
Very interesting, though. 1,000 people.
Yeah, 1,000 plasma. 1,000 donors contribute to the plasma.
That's amazing.
Yeah,
it's amazing science and technology.
It's crazy. But we do use it, but it only works in about 50% because perhaps that's because we're not using it at the right time. We don't really know the proper time to use it. Alongside IVIG, then, if that doesn't work, then we would move to FMT.
Got you.
Yeah.
What's your vision for the future for microbiome therapy and for FMT? Like, where do you think we're going? Like IBD, do you ever think about it for your UC patients, for example? Like, how will that progress over time, do you think?
Yeah, it's difficult. I mean, I think if we're looking at how mature the field is at the minute, it feels very mature for C. diff and well accepted. It's much more stringent to other conditions beyond C. diff, though, and very stringent to conditions that are extraintestinal, because we still need the clinical trial data. On the plus side, there are hundreds of clinical trials now registered on clinicaltrials.gov for FMT, so that's really good news. So hopefully we will get more robust data sets coming through in the next 3 to 5 years. And I think that will inform what we do with FMT. It'll probably inform how we develop precision therapies around the mechanistic value of those samples in the clinical trials. So that, I think, will all, naturally progress and so on. But I still think FMT is not going to disappear anytime soon because, to generate these precision therapies takes absolutely years and years and lots of cash. And so it's not going to happen any quicker than a standard drug.
No.
You're still looking at the same timeline. And only if you want to compress that timeline, then you need a pandemic, basically. You're not going to compress without huge collective resources.
Yeah. And pressure, right?
And pressure.
To find a solution.
Pressure to find a solution. So, I think where it might come into its own a bit is with the multi-drug resistant organisms. If that really is a major cross-council agenda.
Yep.
So we may see more work on that. But as regards, non-communicable diseases, it's a very complex setup, and I am not as confident about that aspect. I think, yes, for UC, that would be sensible on a case-by-case basis,
right?
Plus or minus the clinical trials.
Yeah.
But for Crohn's, the evidence has been very poor.
It's not as good.
Pouchitis, it hasn't been wonderful. So for UC, there's 30 to 40% hit in terms of the meta-analysis for FMT and UC. It's not bad, but it's not
pretty good.
It's pretty good as far as you look at the general thing. But Yeah, I think it will evolve, and you will see more live biotherapeutic products for sure. The problem for us in NHS at this minute in time of austerity is these need to be financially, workable, financially viable. Voust and Rebiota, great to see them being approved by FDA, but between 7 and 17,500 is just not a model that NICE would approve.
No, I completely agree with that. One of the things I just wanted to pick up on there on the IBD was I often wonder, what would happen, do you think, if you had the FMT plus the best-in-class biologic and immune? Like, what do you think? There's maybe a stacking effect associated with all of that?
Yeah, I think now even there have been some clinical trials testing more than one biologic. So combined biologic therapy seems to be, getting a little bit of press attention. So combining biologics, something to definitely look at. And also certainly in the context of FMT, people have combined it with anti-inflammatory diet, for instance, or specific fiber-based diet. So those trials exist, but they're very few and far between. But I do think, yes, there is room to combine FMT with other small molecule therapies, for instance. That would be super interesting as an adjunctive agent.
Yep.
So, you really want to be able to reset things immunologically, and it may not take with the engraftment, but it may take if then you have a supportive adjunctive therapy alongside it. This could really push it over the limit, because I think people have at the minute given FMT or fecal filter, but they haven't given two of them together, or they have, or they haven't given FMT and a metabolite yet,
or a prebiotic,
They haven't done it yet. So those trials need to be done.
And on the subject of prebiotics, do you recommend any prebiotics, probiotics, polyphenols, any supplements to your patients if they come in and say, doctor, what should I be taking for my microbiome?
No. So normally all I say is, can you stop drinking 8 liters of Coca-Cola each day, please? And they just go, really? Do I have to? Yeah, you really do have to. Yeah, because you're just going to float away like a balloon. So yeah, the bloating will certainly go if you stop drinking 8 liters of Coca-Cola each day. So I usually start with the most obvious things that I know they shouldn't do. And then I say, the evidence is not fantastic, but if you look at the different diets, the anti-inflammatory Mediterranean type diet seems to get a lot of traction.
Yeah,
so I tend to just move them in the direction of, the Mediterranean Italian type diet and say, it's tasty. Also, it's good for you as long as, you control portion sizes.
Yeah. And the red wine, is that?
The red wine, absolutely. A little bit of red wine is fine. Plenty of olive oil, And so, we can, and we joke actually, because my colleagues were like, which is the best olive oil, Greek olive oil or Italian olive oil? And I'm just like, oh my God, does it really matter? But these are the things that you want to be able to say to your patient, because there's some evidence backing that up. Prebiotics, we just don't even get there. And as far as probiotics are concerned, all I said to them is they're not going to do you any significant harm, but you need to be buying them from a reputable supplier.
Absolutely.
And if you buy them, it's like herbal remedies. A herbal remedy can cause liver failure. So I'm like, please don't just consume these in masses. You might want to try them if you're going, if you're going somewhere where there's a likelihood of infectious diarrhea. Maybe you'll take them as preventative, something like that. But I tend to not. I tend to say to them, I don't think these are really good. They're not drugs in the sense that they're curing a clinical disease. They're like food supplements. So you kind of have to separate out their expectations and just say to them, I don't think it's going to really do anything for you clinically, but sometimes it's like a good placebo even, sometimes
Absolutely.
They just need something else.
Mm-hmm. And if there was a company that was delivering a Faecalibacterium prausnitzii with stability at room temperature, would you recommend that to your patients, or would you be still cautious?
I would still be cautious because yes, there's a lot of evidence around that particular microbe, but it's usually a consortia, isn't it? It's not just one microbe. There are microbe interactions. So I would tend to say to them, it's a bit like Lactobacillus bifido. Individually, probably not a lot of good for you, but combined with other combined with other microbes that interact and cooperate nicely with each other, yes, that would work potentially, but not on its own. No.
Yeah. That makes sense. So before we wrap up, because we've been talking for an hour now, believe it or not, it does tend to, it flies by, doesn't it?
Yeah.
It really flies by. I just wondered if you had any insights into the GLP-1s and whether the microbiome can contribute to weight loss or weight gain. Because we mentioned at the start, has the microbiome bubble burst?
Yeah.
Yes, no. And I think in some respects, there's a bit of hype, but there's also a lot of great research going on as well at the moment. One of the things that I think triggered the hype was it could be involved in making you more or less overweight. Yeah. Is there any evidence for that?
I think at the minute, no, I don't think so. I mean, certainly some people do benefit metabolically from some of these weight loss drugs, there's no doubt. So if they're in the category of elevated BMI and they've got cardiovascular risk factors, then yes, if they're eligible, they should strongly be considered. And, that improves, it decreases pancreatic inflammation, it increases decreases insulin resistance, all these things that we need to see, especially in diabetic subjects, then absolutely, that's really important. Whether they have a tremendous effect on the microbiome, they probably do. I mean, most drugs do have an impact on the microbiome. But, a lot of the drugs that we see have an impact are PPIs and things like that, antacids. Yeah. What effect they have specifically is difficult to know on its own, never mind in conjunction with other drugs that these individuals will invariably be on. So I think the problem is not necessarily one drug. The problem is usually a polypharmacy effect on the microbiome. So we really have to separate out the effect of those individually versus the polypharmacy effect of other drugs that may even have a more detrimental effect. Or they might also change the pharmacokinetics of those drugs themselves. Those other drugs are on. It may or may not increase their efficacy. Yeah.
It's an independent variable.
It's an independent variable, but patients are desperate to try anything to improve their health. So I would definitely say, the weight loss drugs like Mounjaro and Ozempic and things like that, if there's an indication for you to be on them, then yes, you should take them. But be aware of the caveats that you can get pancreatitis, you can get liver failure. Bad things happen to very few people, but it still does happen.
It still happens. And have you seen any of that in your clinical practice so far?
Yeah, we have seen with the higher doses, we have seen quite significantly deranged liver function tests. But I must say that was really in patients with IBD that I have been managing. Generally speaking, most people have been able to get on with them no problem and escalate their dose.
Yep.
So I think clinically, there's definitely a place for them.
Yeah, I think some of the data looks pretty amazing to see the impact that it's having, both on the weight loss, but also just the broader
health. Yeah, I mean,
pretty amazing.
It is. It's actually quite amazing, and also, to people also changes their appearance and things. They may have tried every diet under the sun,
and they feel amazing as well.
They feel really good.
You see them,
and they're more energetic, and less. I mean, everything is moving in the right direction, and then they don't necessarily have to stay on it lifelong. There's a period of time they would stay on it, and then they'd come off it again.
Yeah. So just a couple of quick fire questions then before I ask you to tell listeners about how they can follow your work and so on. So are there any facts or things about the microbiome that still get you really excited? So someone says, tell me a fact about the microbiome. Is there anything that you are kind of immediately drawn to?
I mean, it kind of changes on a day-to-day basis, doesn't it? I mean, I think the thing I like about it is just the fact that, the majority of the bugs are not culturable. That's the problem. 80 to 90% of metabolites, they're not identified. All we know is some of them are identified, like the short chains and the bile acids, but the vast majority of them we just don't know anything about. So they're just not identified in the scheme of things. And then at least 50% of the genes in our metagenome, we do not know what those genes do. So This is part of the problem, is that there's still an awful lot of mapping to do.
Yeah.
And I think, with the MetaHit and the Human Genome Project, that's done a lot. But that kind of work has to continue so that we have much, much better mapping.
And I think the other one for me today was that there's trace elements of gold.
Oh, yes. Don't forget about the trace. Well, the way I look at it, because I'm kind of like a One Health sort of clinician, so I kind of think, we're just humans at the end of the day, but we live in an environment where there are other animals. So we have to keep thinking about we're sharing, microbial ecosystems, and we have to keep thinking that actually the poo is a resource. So it shouldn't be deemed a waste product.
I like it.
It should be deemed as a resource. So that is how we should just reframe that in our mindsets.
I love that. And that's a perfect way to finish. So thank you so much for coming on. Could you just tell a listener, if they want to follow your work, connect with you, how do they do that?
Best thing is probably just through University of Nottingham. So I have my own internet webpage. So that's probably the best way to connect. Yeah.
Awesome. Well, thank you ever so much for coming on. Really, really enjoyed the conversation. That was super interesting. And wish you all the best and look forward to speaking to you soon.
Thank you so much, James. I appreciate it.